Hello there, and welcome back. I am really glad you are here tonight. If you are near a window, there may be a plant somewhere beyond the glass. A tree along a street, a shrub beside a wall, grass in a courtyard. Perhaps a houseplant is only a few steps away, standing so quietly that it seems almost like part of the furniture. Plants are easy to overlook because they live at a pace that does not match our own. An animal crosses a room in seconds. A bird turns its head. A dog runs toward a sound. A person lifts a hand. Plant movement is usually slower, spread across minutes, hours, days, or seasons. A root changes direction underground where no one sees it. A stem bends toward morning light by fractions of a degree. A flower closes at dusk. A bud forms in autumn and waits through winter. A tree may spend centuries making movements that, from the perspective of a human afternoon, look like stillness. But stillness is not an activity. Inside every living leaf, membranes are maintaining electrical and chemical gradients. Water is moving through narrow channels. Sugars are being loaded into transport tissue. Cells are sensing light, temperature, gravity, touch, water status, pathogens, and chemical signals. DNA is being copied in growing tissues. Proteins are being made and dismantled. Chloroplasts are adjusting to changing light. Mitochondria are releasing usable energy from sugars. Hormones are moving from one part of the body to another. Roots are exploring patches of soil that may never be exposed to air. A plant has no brain, no nerves, and no central control room. Its coordination is distributed. Each region responds to local information while sharing chemical and electrical signals with the rest of the body. The result is not thought in the animal sense. It is something quieter and older, a living system that continuously changes itself in response to the world. Tonight, we will follow that quiet activity from sunlight to leaf, from leaf to root, from root to fungus, from flower to seed, and from one growing season into timescales long enough to be written into wood. We will meet plants that trap insects, plants that wait underground for fire, plants whose flowers deceive pollinators, plants that clone themselves across landscapes, and trees that were already old before many human civilizations began. And because plant science is full of stories that sound almost too beautiful, we will also be careful. Plants do send chemical signals. They do share relationships with fungi. They do respond to touch and vibration. But they do not need to be turned into miniature people in order to be remarkable. Their biology is interesting enough on its own. The deepest story begins long before flowers, forests, or roots. More than 470 million years ago, the continents looked very different. Complex life already filled the oceans, but the land was still comparatively bare. Microbial communities occupied wet surfaces. Fungi were present. Small photosynthetic organisms lived where moisture allowed them to survive. Then one lineage related to freshwater green algae began the long transition toward what we now call land plants. That transition was not one single step. It was an accumulation of solutions to problems that water had previously solved for free. In water, an organism is surrounded by the medium from which it acquires water. On land, water can disappear. In water, the body is supported by buoyancy. On land, gravity pulls every structure downward. Aquatic reproductive cells can move through liquid. On land, reproduction must often continue even when no film of water connects one individual to another. Sunlight can be stronger. Temperatures can change faster. Nutrients are locked inside rock and soil rather than dissolved around the body. Early land plants evolved ways of reducing water loss, protecting reproductive cells, and surviving drying. A waxy outer coating, the cuticle, helped slow evaporation. Spores developed resistant walls. Openings that later became the sophisticated stomata of modern plants allowed gas exchange while limiting uncontrolled water loss. Partnerships with fungi may have helped early plants acquire mineral nutrients from the primitive soils of the time. They were not trees. They did not begin with roots stretching meters underground. The first land plants were small, and their living descendants should not be mistaken for unchanged fossils of those ancestors. Modern mosses, liverworts, and hornworts have had hundreds of millions of years of evolution of their own, but they preserve something of the scale of a world before vascular tissue allowed plants to rise high above the ground. The development of vascular tissue changed that scale dramatically. Xylem made long-distance water transport possible. Phloem made long-distance transport of sugars and other compounds possible. Lignin strengthened cell walls. Roots penetrated deeper into soil. Leaves created broad surfaces for collecting light. Plants could now build bodies far larger than diffusion alone would allow. As plants spread across continents, they changed the continents themselves. Roots fractured rock. Organic matter accumulated in soils. Photosynthesis removed carbon dioxide from the atmosphere. Plant tissues fed fungi, microbes, and animals. Forests altered the movement of water between ground and atmosphere. The colonization of land by plants did not simply add greenery to an existing world. It helped create the terrestrial world that later animals inherited. To understand how, we can move inward from an entire forest to one living cell. A plant cell is enclosed by a plasma membrane, as animal cells are, but outside that membrane lies a cell wall. The wall is built mainly from cellulose microfibrils embedded in a matrix of other carbohydrates and proteins. Cellulose itself is made from glucose molecules linked in a way that human digestive enzymes cannot break. It is extraordinarily abundant. Much of the dry structure around us in wood, paper, cotton, and plant fiber is cellulose produced by living cells. The wall gives a plant cell something animal cells generally do not have: the ability to resist high internal pressure without bursting. Water enters the cell by osmosis. A large central vacuole takes up much of the interior volume. As water accumulates, the contents of the cell press outward against the wall. The wall pushes back. This turgor pressure helps keep soft stems and leaves firm. A herbaceous plant can stand upright without a skeleton because billions of pressurized cells act like tiny structural units. When water becomes scarce, the pressure drops. Leaves become limp. Stems bend. The plant wilts not because its cellulose has suddenly vanished, but because the hydraulic support inside its living cells has weakened. Neighboring plant cells are not completely isolated by their walls. Tiny channels called plasmodesmata cross the walls and connect the cytoplasm of adjacent cells. Through these channels, small molecules and signals can move from cell to cell. A plant body is divided into cells, yet those cells remain chemically connected in ways that allow tissues to behave as coordinated systems. And then there are the chloroplasts. A chloroplast looks like an organelle, but its evolutionary history reaches back to an ancient encounter between entirely separate organisms. More than 1 billion years ago, an ancestral eukaryotic cell formed a lasting partnership with a photosynthetic cyanobacterium. Instead of being digested, the cyanobacterium remained inside. Over evolutionary time, most of its genes were lost or transferred to the host nucleus. The once independent organism became an organelle. A chloroplast still carries traces of that origin. It has its own small genome. It is surrounded by membranes. It divides from pre-existing chloroplasts, and inside it lies the machinery that connects life on land to sunlight. Imagine a photon arriving from the sun. It left the solar surface roughly 8 minutes ago. It crosses space, enters Earth's atmosphere, passes through the leaf surface, and reaches a pigment molecule in a chloroplast. Chlorophyll absorbs some wavelengths more strongly than others, especially in the blue and red portions of visible light. Green wavelengths are more likely to be reflected or transmitted, which is one reason many leaves appear green to human eyes. Absorption raises an electron in the pigment to a higher energy state. That event lasts an almost unimaginably short time, yet it begins a chain of reactions that can eventually contribute to a sugar molecule. The light-dependent reactions of photosynthesis occur in internal chloroplast membranes called thylakoids. Proteins, pigments, electron carriers, and enzymes are arranged there with remarkable precision. In photosystem 2, light energy helps remove electrons from water. The water molecules are split. Their electrons enter the photosynthetic electron transport chain. Hydrogen ions contribute to a gradient across the membrane. Oxygen is released. That oxygen is chemically important, but from the plant's point of view, it is not the main product being sought. It is a consequence of obtaining electrons from water. The electrons move through a series of carriers. Their energy is used to pump protons across the thylakoid membrane. The resulting concentration gradient powers ATP synthase, a rotating molecular machine that produces ATP. In photosystem 1, another photon raises the energy of the electrons again, helping generate NADPH. ATP and NADPH are temporary packages of chemical energy and reducing power. They are then used in the Calvin cycle, where the plant begins incorporating carbon dioxide into organic molecules. The enzyme Ribisco attaches carbon dioxide to a 5-carbon molecule. The intermediate immediately splits. Through a sequence of reactions, carbon is rearranged into 3 carbon compounds that can eventually contribute to sugars, starches, cellulose, amino acids, oils, pigments, and thousands of other molecules. This process is often summarized by a simple equation in textbooks. Carbon dioxide and water, using light energy, become sugar and oxygen. The equation is correct as a summary. Inside a leaf, however, the reality is a coordinated landscape of membranes, ions, enzymes, pigments, transport proteins, and competing reactions. Ribisco, one of the most abundant enzymes on Earth, is not perfectly selective. It can bind oxygen instead of carbon dioxide. When it does, the plant enters a costly pathway called photorespiration. Under hot, dry conditions, the problem becomes more serious because stomata tend to close to conserve water, reducing the carbon dioxide concentration inside the leaf while oxygen remains. Evolution has produced several ways of managing this trade-off. Most plants use what is called C3 photosynthesis, named for the 3 carbon products formed early in carbon fixation. Many grasses from warm climates, including maize and sugarcane, use C4 photosynthesis. They first capture carbon dioxide in 4 carbon compounds and concentrate it around Ribisco in specialized cells. This reduces photorespiration and can improve performance under hot, bright conditions. Cam plants use another solution. Cacti, many agaves, pineapples, and numerous orchids open their stomata mainly at night. They take in carbon dioxide when temperatures are lower and the air may be more humid, storing the carbon temporarily in organic acids. During the day, the stomata can remain more closed while the stored carbon dioxide is released internally for photosynthesis. This is a useful reminder for later tonight. Not every plant follows the same daily schedule. A forest at night is quieter in one sense because light-driven photosynthesis has stopped, but some plants are opening their stomata precisely because darkness has arrived. Even within one ordinary leaf, the architecture reflects compromises. A leaf needs light, but too much light can damage photosynthetic machinery. It needs carbon dioxide, but every open stoma allows water vapor to escape. It needs to be broad enough to capture sunlight, but thin enough that gases and heat can move efficiently. It must resist pathogens, herbivores, wind, drought, and ultraviolet radiation while remaining cheap enough to build that the plant can replace it. Many leaves contain a palisade layer near the upper surface, where tightly packed cells are rich in chloroplasts and well-positioned to capture light. Below lies more loosely arranged spongy tissue with air spaces that help carbon dioxide diffuse through the leaf. Veins carry water inward and sugars outward. Stomata, often concentrated on the lower surface, regulate exchange with the atmosphere. Guard cells control each stomatal pore. When they accumulate ions, water follows osmotically, changing cell pressure and shape so the pore opens. When signals such as drought-related abscisic acid trigger ion release, water leaves the guard cells and the pore narrows. This opening and closing can happen thousands or millions of times across one plant. No leaf is simply a solar panel. It is a regulated exchange surface, continuously balancing carbon gain against water loss. And because water is constantly being lost to the air, we can now follow the next quiet wonder of plant life downward, into the plumbing of a stem and the dark soil beneath it. A tall tree seems to present a simple mechanical problem. The roots are in the soil. The leaves may be tens of meters above them. Water must somehow travel from one to the other. Animals solve long-distance fluid transport with pumps. Hearts contract. Blood is forced through vessels under positive pressure. Trees have no equivalent central pump. The movement of water through xylem depends mainly on physical forces generated by evaporation from leaves. When water evaporates from moist cell walls inside a leaf and diffuses through the stomata, the remaining water is pulled toward those evaporating surfaces. Water molecules attract one another through hydrogen bonding. They also adhere to the walls of narrow xylem conduits. Because a continuous column of water connects leaves to roots, tension created near the top can be transmitted downward through the column. The water in functioning xylem is therefore often under negative pressure. It is being pulled rather than pushed. This cohesion tension mechanism allows a tree to lift enormous quantities of water without using metabolic energy to pump each molecule upward. The sun provides much of the energy indirectly by driving evaporation. The scale becomes especially impressive in the world's tallest trees. Coast redwoods can rise beyond 100 meters. Hyperion, a coast redwood in California, has been measured at a little over 116 meters. At its highest living leaves, the hydraulic path from soil to canopy is longer than the height of many city buildings. There are limits. As tension in xylem becomes more negative, the water column becomes vulnerable to cavitation. A tiny gas bubble can expand and block a conduit, creating an embolism. Drought increases the risk because the plant must pull harder to extract water from drying soil. Freezing can also create bubbles when dissolved gases come out of solution. Plants have evolved different xylem architectures that trade efficiency against safety. Wide vessels can conduct water quickly, but may be more vulnerable to some forms of failure. Narrower conduits can be safer, but move less water. Some plants can isolate damaged vessels. New xylem can be produced as stems grow. In certain cases, embolized conduits may regain function, though the importance and mechanisms of refilling under tension are still studied. The hydraulic problem helps explain why the tallest trees have a practical height limit. Gravity matters. Path length matters. Friction matters. At great heights, leaves become smaller and water stress increases even when the soil below is wet. Coast redwoods receive an additional gift from their environment: fog. Moist coastal air can reduce the rate of water loss from leaves, and water from fog can wet foliage and enter ecosystems directly. The highest parts of a redwood are therefore connected not only to soil water below, but also to atmospheric moisture around the canopy. Water is one half of the vascular story. Sugar moves through another tissue. Phloem carries products of photosynthesis from sources to sinks. A mature sunlit leaf may be a source because it produces more sugar than it needs immediately. A growing root tip, developing fruit, expanding young leaf, flower, storage organ, or seed may be a sink because it consumes or stores sugar. Sugar, often transported mainly as sucrose, is actively loaded into phloem tissue in many species. Water follows osmotically from nearby xylem, increasing pressure. At a sink, sugar is removed and water potential changes. The resulting pressure differences drive bulk flow through the phloem. A tree, therefore, contains two great transport systems operating according to different physics. Xylem carries mostly water and minerals, usually from roots upward, under tension. Phloem carries sugars and many signaling molecules from sources to sinks under pressure, and its direction can differ in different regions of the plant depending on where resources are being produced and used. During spring, a storage organ can become a source. In autumn, leaves that were sources may send resources into roots and stems before they are shed. A developing fruit can pull sugar from several leaves. A shaded branch may depend partly on resources produced elsewhere. The plant's body is a shifting map of supply and demand. Now we can follow the water column all the way down. A root tip advances through soil without knowing in advance what lies ahead. Near its very end is a root cap that protects delicate growing tissue as it pushes between mineral particles. Behind the cap lies a region where cells divide, followed by a region where newly produced cells elongate. Farther back, cells differentiate into specialized tissues. Root hairs emerge from epidermal cells and dramatically increase the surface area in contact with soil. Each root hair is tiny and short-lived, but large populations are continually replaced as the root grows. Soil is not a uniform reservoir. A few millimeters can separate a moist pore from a dry one, a phosphate-rich particle from a depleted region, or a zone filled with oxygen from one where waterlogging has driven oxygen away. Roots respond to this patchiness by changing growth and branching. Gravity provides one of the most reliable directional cues. Specialized cells in root caps contain dense starch-filled organelles called statoliths. These settle in the direction of gravity, helping the root reorient growth downward. Shoots use related gravity-sensing mechanisms, but usually bend in the opposite direction. Roots also respond to moisture gradients, nutrient availability, oxygen, physical obstacles, neighboring roots, microbes, and chemical conditions. Hydrotropism can bias growth toward water. Nutrient-rich patches can stimulate local branching. Hard objects can redirect the tip. Toxic concentrations of salts or metals can change growth. The final pattern is not a fixed blueprint. It is an architecture built through continuous exploration. One of the most famous measurements of root complexity came from early 20th century work in which whole root systems were painstakingly excavated and mapped. A single cereal plant could produce hundreds of kilometers of fine roots when all branches were added together, and the contribution of root hairs made the absorbing surface vastly greater still. The exact numbers depend strongly on species, soil, growing conditions, and measurement method, so the deeper lesson is not one spectacular statistic. It is that most of a plant's contact with the world may be hidden beneath the ground, and roots rarely explore that world alone. Immediately around a root lies the rhizosphere, a narrow zone profoundly influenced by the plant. Roots release sugars, amino acids, organic acids, mucilage, ions, and many other compounds. Some leak passively. Others are secreted in ways that alter the chemistry and biology of the surrounding soil. These substances feed microbes. Bacteria gather around root surfaces. Fungi grow along and inside roots. Protozoa consume bacteria. Tiny animals graze on fungi and microorganisms. Pathogens attempt to enter. Beneficial microbes compete with them. Mineral nutrients are dissolved, immobilized, released, or transformed. A root is therefore not simply a drinking straw inserted into dirt. It is the center of a living chemical neighborhood. Among the most important relationships in that neighborhood are mycorrhizas. The word comes from Greek roots meaning fungus and root. In a mycorrhizal partnership, fungi colonize the plant's root system while fungal filaments, called hyphae, extend outward into soil. These hyphae are much thinner than most roots and can enter tiny pores that roots cannot reach efficiently. The fungal partner acquires mineral nutrients and water. The plant supplies carbon compounds produced by photosynthesis. The exchange can be enormously important, especially for phosphorus, nitrogen, and other nutrients that diffuse slowly or occur in chemically inaccessible forms. Different mycorrhizal systems are built differently. Arbuscular mycorrhizal fungi enter root cortical cells and form highly branched exchange structures called arbuscules. Ectomycorrhizal fungi form a sheath around fine roots and a network between root cells without normally entering the cells themselves. Orchids form specialized mycorrhizal relationships, especially during germination, when their tiny seeds have almost no stored food and depend on fungal carbon. Many heathers and relatives form ericoid mycorrhizas adapted to nutrient-poor acidic soils. The exchange is not a sentimental friendship. It is a biological trade whose terms change. A plant may direct more carbon toward fungal partners that provide more nutrients. Fungi can allocate resources among hosts. The value of the partnership changes with soil fertility, water, species identity, and environmental stress. Under some conditions, the cost of supporting a fungus may approach or exceed the benefit it provides. Because fungal hyphae can colonize more than one plant, roots can become linked in common mycorrhizal networks. Tracer experiments have shown that carbon and other materials can move between connected plants in some circumstances. This is where a careful distinction matters. The existence of fungal connections is well established. Resource movement through them has also been measured. But popular descriptions sometimes go much farther, portraying forests as if large trees intentionally feed their offspring, send purposeful warnings through an underground internet, or manage neighboring plants through a cooperative social network. Those stronger claims are not established in the same way. Field studies are difficult to interpret because materials can also move through soil, microbes, roots, and other pathways. The ecological importance of transfer varies. Whether transferred carbon meaningfully improves the recipient's survival in natural forests is still debated. The real story does not become less interesting when intention is removed. A forest floor contains networks formed by organisms with different evolutionary interests. Molecules move through them. Nutrients are exchanged. Pathogens and competitors interact. Seedlings can be influenced by the fungi already associated with adult trees. The network changes which organisms can reach which resources. That is already extraordinary. Plants can also influence one another without fungal connections. When leaves are chewed, damaged, or infected, they often release volatile organic compounds into the air. Neighboring leaves on the same plant, and sometimes neighboring plants, can detect parts of this chemical mixture and alter defensive responses. Depending on the species and experiment, receiving tissues may increase defensive enzymes, change hormone signaling, or become primed to respond more quickly when attack arrives. Some volatiles attract predators or parasitoids of the herbivore. A damaged plant can therefore change the behavior not only of another plant, but of an insect searching for another insect. Again, the word communication can be useful if we define it carefully. Information is moving because one organism's state changes a signal and another organism responds to that signal. It does not imply conscious sending, conscious receiving, or a language with meanings like human words. Plants have no need for those additions. Their chemical world is rich enough. To understand how a leaf detects that it has been bitten, we need to look at another form of quiet activity, plant immunity. A plant cannot run from infection. If a fungus enters a leaf, if a bacterium reaches damaged tissue, or if a virus begins copying itself inside a cell, the threatened plant must respond where it stands. Plants do not have circulating immune cells like neutrophils or lymphocytes. They do not make antibodies. Their immune system is built largely into individual cells and reinforced by signals that spread through tissues. The first layer is physical. Waxy cuticles make leaf surfaces difficult to penetrate. Cell walls form mechanical barriers. Stomata can close when danger is detected. Bark, cork, hairs, antimicrobial compounds, and the microbiome on plant surfaces all reduce the chance that a pathogen reaches vulnerable living cells. When pathogens do enter, plant cells recognize characteristic molecules. Cell surface receptors can detect conserved fragments of microbial structures, such as bacterial flagellin or fungal chitin. Recognition activates signaling pathways. Calcium concentrations change. Reactive oxygen species are produced. Protein kinases alter the activity of other proteins. Defensive genes are turned on. Cell walls may be reinforced. Antimicrobial compounds accumulate. Successful pathogens evolve molecules called effectors that suppress these defenses or manipulate host cells. Plants, in turn, evolve intracellular immune receptors that can detect some effectors directly or detect the changes they cause. This creates an evolutionary contest that can be remarkably specific. When strong recognition occurs, cells near the infection may sometimes undergo a localized form of death called the hypersensitive response. Sacrificing a small region can restrict pathogens that require living tissue. The damaged patch may become visible as a tiny brown lesion. Signals also move beyond the infection site. One important pathway involves salicylic acid and leads to systemic acquired resistance. After a local infection, distant parts of the plant can enter a primed defensive state. They are not necessarily producing every defense at maximum level all the time. Instead, they become prepared to respond more rapidly or strongly when a later challenge appears. The plant therefore enters a whole body state of immune priming. The effect can persist after the first infection, but it is not antibody-based memory like the adaptive immunity of vertebrates. The response depends on the kind of threat. Salicylic acid pathways are often important in resistance to pathogens that feed on living cells. Jasmonate and ethylene signaling are especially important in many responses to chewing herbivores and pathogens that kill tissue. These pathways interact, sometimes strengthening and sometimes suppressing one another. The plant must choose carefully because defense is expensive. Carbon used to make toxins cannot be used simultaneously to make more leaf area. Nitrogen invested in defensive proteins cannot also build photosynthetic enzymes. Closing stomata may block a pathogen, but it also limits carbon dioxide entry. Killing infected cells can stop one disease and create damaged tissue. Plant defense, like animal immunity, is an exercise in proportionality. Many of the chemicals produced in this struggle have become familiar to humans for completely different reasons. Caffeine is a plant alkaloid. So is nicotine. Capsaicinoids make chili peppers feel hot to mammals. Tannins create astringency. Terpenes help produce the scents of pine, mint, rosemary, citrus peel, and countless flowers. Salicylate chemistry contributed to the development of aspirin. Alkaloids from opium poppy gave medicine morphine and codeine. Compounds from cinchona bark led to quinine. Paclitaxel, first isolated from Pacific yew bark, became an important anti-cancer drug. It is tempting to imagine that plants made these compounds for us. They did not. Each molecule arose within the plant's own ecology. Some deter herbivores. Some inhibit microbes. Some attract pollinators. Some protect against ultraviolet light or oxidative stress. Some affect competing plants. Some may serve several functions at once. Humans entered these chemical relationships very late and learned to taste, extract, cultivate, modify, and manufacture what evolution had already produced. A single plant can make hundreds or thousands of specialized metabolites, and the mixture can change after injury. A caterpillar begins chewing a leaf. Damaged cells release signals. Fatty acid derivatives contribute to jasmonate production. Defensive genes become active. Proteinase inhibitors may make digestion more difficult for the herbivore. Toxic or unpalatable compounds may increase. Volatile compounds drift into the air. Sometimes the attacker itself contributes information. Molecules in insect saliva can help the plant distinguish chewing from simple mechanical damage. The plant is not merely responding to a hole. It is interpreting molecular evidence about how the hole was made. This capacity to distinguish types of disturbance brings us to the broader question of what plants can sense. Light is one of their richest sources of information. Plants do not have eyes, but they possess several families of photoreceptors tuned to different wavelengths. Phytochromes respond strongly to red and far-red light. Cryptochromes and phototropins respond to blue light. UV-8 detects ultraviolet B. These receptors allow a plant to use light not only as energy, but as information about the environment. Consider a seedling emerging from soil. Underground, its developmental program keeps the stem elongated and the leaves relatively undeveloped. When it reaches light, photoreceptors trigger a dramatic transition. The stem changes its growth pattern. Cotyledons, or leaves, expand. Chloroplasts mature. Photosynthetic genes become active. Light has told the seedling that it has reached the surface. The ratio of red to far-red light carries another message. Leaves absorb red light strongly for photosynthesis and transmit or reflect more far-red light. A plant beneath neighboring foliage therefore experiences a lower red to far-red ratio than a plant in open sunlight. Phytochromes detect this difference. Many species respond with shade avoidance growth. Stems or petioles elongate. Leaves change orientation, and resources are directed toward reaching brighter conditions. No plant has looked upward and decided that another plant is blocking the sun. A receptor has changed molecular state. A signaling network has altered gene expression, and growth has been redirected. Yet from outside, the result can resemble purposeful competition. Phototropism creates another familiar movement. When light reaches one side of a young shoot, blue light photoreceptors help redistribute the hormone auxin. Cells on the shaded side elongate more than cells on the illuminated side. The stem curves toward the light. Charles Darwin and his son Francis studied this response in grass seedlings in the 19th century. They found that covering the tip prevented bending even when the lower region remained illuminated. The tip somehow detected light and transmitted an influence downward to the region that bent. They could not yet know the molecular identity of auxin. Their simple experiments revealed a central principle of plant development. Sensing and growth can occur in different places. Auxin became one of the major plant hormones. It influences cell expansion, root development, vascular patterning, fruit growth, branching, and many other processes. Its effects depend on concentration, tissue, transport direction, developmental stage, and interactions with other hormones. Auxin is transported in a highly organized way from cell to cell by membrane proteins whose positions can themselves change. This directional transport allows local auxin maxima and gradients to form. Those patterns can help decide where a new leaf appears, where a lateral root begins, or which side of a stem elongates more rapidly. Other hormones participate in the same distributed coordination. Cytokinins influence cell division, shoot growth, nutrient signaling, and leaf aging. Gibberellins can promote stem elongation and seed germination. Abscisic acid is central to drought responses and seed dormancy. Ethylene, a gas, regulates fruit ripening, senescence, flooding responses, and growth around obstacles. Jasminates coordinate many responses to wounding and herbivores. Brasinosteroids regulate growth and development. Strigolactones influence shoot branching and also leak into soil, where they help attract arbuscular mycorrhizal fungi. The boundaries between hormone functions are not neat. A plant does not have one hormone for growth and another for stress. Growth is stress-dependent. Defense changes development. Water status changes flowering. Light changes hormone transport. Nutrient availability changes root architecture. The plant's shape is an ongoing calculation performed through chemistry. Touch also changes that shape. A climbing vine that contacts a support can curve around it. Tendrils coil. Roots redirect around barriers. Trees exposed to persistent wind often grow shorter and sturdier than genetically similar trees in sheltered locations. This long-term response to mechanical stimulation is called thigmomorphogenesis. Some touch responses are much faster. The sensitive plant, Mimosa pudica, folds its leaflets and droops after mechanical disturbance. The movement depends on rapid changes in ion concentrations and water pressure in specialized structures called pulvini. Electrical signals can spread through the plant, helping coordinate the response. The Venus flytrap is faster still. Its trap is a modified leaf with sensitive trigger hairs on the inner surface. A single touch is usually not enough to commit the plant to closure. The plant effectively counts electrical events over a short interval. If trigger hairs are stimulated again soon enough, action potentials spread and the trap snaps shut. The motion is not a simple muscle-like contraction. Elastic energy stored in the curved leaf and rapid changes in geometry allow the lobes to switch between stable shapes. After capture, additional stimulation by struggling prey helps trigger a tighter seal and digestive responses. This counting mechanism reduces the cost of closing for raindrops or random debris. A trap that shuts unnecessarily loses time and energy. A trap that never responds loses prey. Selection has shaped a threshold. Carnivory has evolved independently many times among flowering plants, not merely once or twice. Different lineages have transformed leaves into pitfall traps, sticky surfaces, suction bladders, snap traps, and other structures. Why would a photosynthetic organism eat an animal? Usually not for energy. The plant still obtains most of its energy from light. What the animal provides is a concentrated source of scarce mineral nutrients, especially nitrogen and phosphorus. Carnivorous plants often thrive in bright habitats where water may be abundant but soils are acidic or nutrient poor. A sundew spreads glistening droplets of sticky mucilage across tentacles. Small insects become trapped. Tentacles, and sometimes the leaf, slowly bend inward. Digestive enzymes release nutrients. Pitcher plants create deep cavities with slippery surfaces, downward-pointing structures, nectar, scents, or visual cues that guide insects toward the rim. Prey falls into fluid where it is digested by enzymes, microbes, or both. Bladderworts, which often live in water, use tiny traps held under negative pressure. When a small organism touches trigger hairs near the entrance, a door opens and water rushes inward, carrying the prey with it. The event can occur in only a few milliseconds, making it one of the fastest movements known in plants. There are even carnivorous bromeliads and lineages with underground traps for microscopic soil organisms. The forms differ because carnivory is a repeated evolutionary solution assembled from ordinary plant machinery. Genes involved in defense, nutrient transport, secretion, and leaf development can be repurposed into traps and digestive systems. Plants did not have to invent an entirely new biology. Evolution modified what was already there. The same principle applies to parasitic plants, which move in the opposite direction. Instead of capturing animals for minerals, they tap directly into other plants. Daughter seedlings emerge from seeds with only a short window to find a host. Their slender stems grow in searching arcs and respond to chemical cues released by nearby plants. After contact, they coil around the host and form haustoria, specialized structures that penetrate tissues and connect to the host's vascular system. Mistletoes also form haustorial connections, but often remain photosynthetic themselves, stealing water and minerals while producing some of their own sugars. At the extreme are plants such as Rafflesia, famous for enormous flowers and the odor of decaying flesh. Most of the plant lives hidden inside its host as thread-like tissue. There may be no ordinary leaves, stems, or roots. Then, for reproduction, a massive flower emerges. A plant body, we begin to see, is far less fixed than the familiar image of root, stem, leaf, and flower suggests. Evolution has repeatedly reshaped the same basic developmental toolkit, and nowhere is that reshaping more spectacular than in the flower. Before flowers existed, land plants were already reproducing successfully for hundreds of millions of years. Mosses and their relatives release spores. Ferns also disperse spores and produce a separate gametophyte generation. Seed plants introduced another powerful innovation. The embryo could be packaged with protection and stored food, and pollen could carry the male gametophyte without requiring free water to swim between plants. Gymnosperms, including conifers, cycads, ginkgo, and metaphytes, produce seeds that are not enclosed inside fruits. Their reproductive structures can be large cones or much smaller forms. Wind carries enormous quantities of pollen in many species. Then, during the age of dinosaurs, flowering plants diversified. The exact earliest history of flowers is still reconstructed from fossils, molecular evidence, and the relationships among living species. By roughly 140 million years ago, unmistakable flowering plants were present. Over the following tens of millions of years, angiosperms spread into many habitats and eventually became the dominant plant group across much of the world's land surface. A flower is a reproductive structure built from modified organs. Sepals often protect the developing bud. Petals may attract pollinators, though many flowers lack showy petals entirely. Stamens produce pollen. Carpels contain ovules. After fertilization, ovules can develop into seeds, and the surrounding ovary can become fruit. The remarkable part is not simply that flowers reproduce. Cones reproduce too. The remarkable part is how flowers opened an immense evolutionary space for relationships with animals. A pollinator does not visit a flower because the plant needs help. It visits because something in the flower matters to the pollinator. Often that something is nectar, a sugar-rich liquid. Sometimes it is pollen itself, rich in protein and other nutrients. In other cases, the flower offers oils, fragrances, shelter, heat, or even deception without a real reward. Natural selection acts on both sides. A flower that attracts an efficient pollinator leaves more descendants. A pollinator that finds flowers efficiently gains more food or other resources. Over generations, colors, scents, shapes, timing, and behavior can become closely matched. Human vision sees only part of this conversation. Many bees can see ultraviolet wavelengths that humans cannot. Some flowers contain ultraviolet patterns that form guides toward nectar and pollen. To us, a petal may look uniformly yellow. To a bee, it may contain a dark central target. Bird-pollinated flowers often emphasize colors that birds see well and may produce large amounts of dilute nectar. Their shapes can accommodate hovering hummingbirds or perching visitors. Flowers pollinated by nocturnal moths are often pale, strongly scented, and most fragrant after sunset. Bats pollinate numerous tropical plants. Their flowers may open at night, produce strong odors, offer large amounts of nectar and pollen, and be positioned where a flying mammal can reach them. Some flowers are heated. Certain arums and other plants use metabolic energy, to raise the temperature of floral tissues. Heat can help volatilize odor compounds and may provide a warm microenvironment for visiting insects. Some flowers smell like fruit. Others smell like honey. A few smell like carrion or dung because the insects they need are searching for those materials. Evolution does not care whether a perfume is pleasant to humans. One of the most elegant stories of prediction in evolutionary biology involves a white orchid from Madagascar. Angraecum sesquipedale produces a flower with an extraordinarily long nectar spur. When Charles Darwin examined it in the 19th century, he reasoned that some moth must exist with a proboscis long enough to reach nectar near the bottom. Such a moth would press against the flower's reproductive structures while feeding and could transfer pollen between flowers. A long-tongued hawk moth capable of the interaction was later documented. Taxonomy and measurements have been refined since Darwin's day, but the central logic remains beautiful. The architecture of one organism can reveal something about the unseen organism with which it evolved. Not every plant-pollinator relationship is mutual. Some orchids deceive. Certain species mimic the appearance and chemical signals of female insects. A male attempts to mate with the flower and leaves carrying pollen. The plant has obtained pollination without paying a nectar reward. Other flowers mimic food sources or brood sites. Some temporarily trap insects and release them later after pollen transfer. And a few plants reverse the usual arrangement so dramatically that they seem almost theatrical. The giant flowers of Rafflesia use color, texture, heat, and odor to resemble decaying tissue, attracting carrion flies that would normally visit an animal carcass. The insect is not fooled because it is unintelligent. It is responding to sensory cues that are normally reliable in its world. The plant has evolved to produce those cues. Flowering also has to occur at the right time. A plant that blooms too early may encounter frost or find no pollinators. One that blooms too late may fail to mature seeds before drought or winter. Plants combine several environmental signals to time the transition. Day length is one of the most reliable because, unlike temperature, it changes predictably with season. Leaves measure the duration and quality of light through photoreceptor and circadian clock systems. In many species, a mobile protein signal produced in leaves travels through phloem to the shoot tip and helps convert a vegetative meristem into a floral meristem. The famous flowering signal is often associated with the protein FT. Decades before its molecular identity was known, researchers proposed a hypothetical mobile substance called phloragen. Modern genetics revealed that the old physiological idea had pointed toward a real long-distance signaling system. Some plants require a period of winter cold before they can flower efficiently. This process, vernalization, prevents them from responding to a brief warm spell in autumn as if spring had arrived. Prolonged cold changes gene regulation, leaving a molecular memory that winter has occurred. A plant can therefore integrate light history, temperature history, age, nutrition, and hormonal state before producing a flower. Once pollination succeeds, another extraordinary process begins. A pollen grain lands on a compatible stigma. It hydrates and grows a pollen tube. The tube extends through female tissue toward an ovule, guided by chemical and cellular signals. Sperm cells travel inside the tube. Flowering plants perform double fertilization. One sperm fertilizes the egg and produces the embryo. Another fuses with other nuclei in the ovule to create endosperm, a nutritive tissue that supports the developing embryo. The seed that forms is neither simply an embryo nor a miniature dormant adult. It is a carefully packaged developmental system. A seed coat protects it. Stored carbohydrates, oils, or proteins provide energy. Hormonal balances control dormancy. The embryo contains embryonic root and shoot tissues ready to resume growth when conditions become suitable. Some seeds can germinate almost immediately. Others wait. Dormancy is one of plant biology's most impressive uses of time. A seed may need to survive one winter, many summers, a forest fire, passage through an animal, or decades beneath soil. Germinating at the wrong moment can be fatal because a seed usually gets only one attempt. Abscisic acid helps establish and maintain dormancy in many seeds. Gibberellins often promote germination. Temperature, moisture, light, nitrate, smoke chemicals, and damage to the seed coat can alter the balance. Some seeds require cold stratification. A period of moist chilling tells them that winter has passed. Some desert seeds respond only after enough rainfall has washed away inhibitors or provided sustained moisture. Certain fire-adapted species respond to heat or chemicals in smoke, allowing seedlings to emerge after fire has opened space, released nutrients, and reduced competition. Hard-coated seeds may remain impermeable to water until abrasion, temperature change, microbial action, or passage through an animal weakens the coat. There are astonishing examples of longevity, but they need to be told carefully. Ancient lotus seeds from dry lake sediments in China have been germinated after roughly a millennium or more, though assigning exact ages to individual seeds has required careful dating. Seeds of the Judean date palm, recovered from archaeological sites, have also germinated after about 2,000 years. The resulting palms demonstrate that, under unusual dry conditions, viable plant embryos can survive across historical eras. An even older story is often told incorrectly. In Siberian permafrost, researchers recovered fruits of Silene stenophylla from deposits roughly 30,000 years old. Whole fertile plants were regenerated in culture from exceptionally preserved maternal tissue inside immature fruits. The ancient seeds themselves were not simply planted in soil and awakened after 30,000 years. The distinction matters because the true result is remarkable enough. Living plants were reconstructed from ancient preserved plant tissue dating to the late Pleistocene. Most seeds, of course, survive nothing like this long. Some remain viable for only days or months. Others form persistent soil seed banks lasting years or decades. Longevity depends on species, moisture, temperature, oxygen, seed chemistry, and storage conditions. When a seed finally commits to germination, its quiet waiting ends quickly. Water enters. Metabolism accelerates. Stored food is mobilized. The embryonic root, the radicle, usually emerges first and anchors the seedling. The shoot follows. Once leaves reach light, the young plant begins shifting from stored reserves toward photosynthesis. The entire adult body may eventually arise from a structure small enough to rest on a fingertip. But before the seed can begin that next life, it has to leave its parent or survive near it. Plants have evolved almost every imaginable solution to the problem of dispersal. Dandelion fruits carry parachute-like structures that increase drag and allow air currents to carry them away. Maples and many other trees produce winged fruits that auto-rotate as they fall. Tiny orchid seeds can drift through air like dust. Coconuts float. Their thick, fibrous husks help protect the seed during transport by water, allowing some to cross marine distances and establish on coasts. Burdocks and other plants attach hooks to animal fur. The burrs that clung to clothing and dog hair inspired George de Mestral to study their structure and eventually develop Velcro. Many flowering plants use a more elaborate bargain, fruit. A ripe fruit contains sugar, water, pigments, aromas, acids, and nutrients that make it attractive to an animal. The animal eats it. The seeds survive chewing or digestion well enough to be transported somewhere else. The color of ripe fruit often reflects the vision of its dispersers. Fruits eaten by birds can remain conspicuous against foliage to avian eyes. Mammal-dispersed fruits may rely strongly on scent. Some fruits change color during ripening. advertising when seeds are mature enough to leave the parent. Hot chili peppers offer a particularly elegant example. Capsaicin strongly activates pain receptors in mammals, discouraging many mammals from eating the fruit. Birds are much less sensitive to capsaicin. Birds also tend to swallow seeds without crushing them and can transport them long distances. The same molecule that gives a human dinner its heat may therefore influence which animals disperse a wild pepper's seeds. Some plants launch seeds mechanically. Touch-me-nots build tension in ripe fruits that burst when disturbed, flinging seeds outward. Witch hazel can eject seeds with considerable speed. The sandbox tree produces explosive fruits with such force that its common stories sometimes sound exaggerated, though explosive dispersal itself is very real. Others rely on animals not merely to eat fruit, but to store seeds. Jays, nutcrackers, squirrels, and other animals cache nuts and seeds for later. They retrieve many but not all. Forgotten caches can become seedlings. Oaks, pines, and other trees have benefited from animal memories that are excellent, but not perfect. Once dispersed, a seed enters a landscape filled with other plants, microbes, weather, and chance. Some seedlings will live for days. Others may begin bodies that outlast generations of humans. To understand that possibility, we can move from the soft tissues of a seedling into wood. A tree trunk is a history of repeated construction. Near the outside of a woody stem lies a thin cylinder of dividing cells called the vascular cambium. Cells produced toward the inside become secondary xylem, which we call wood. Cells produced toward the outside contribute to secondary phloem. Year after year, the cambium adds new conducting and supporting tissue. In climates with strong seasons, the character of that wood changes through the growing season. Early in spring, many trees produce larger, thinner-walled conducting cells suited to rapid water transport. Later, growth often produces denser tissue with smaller cells and thicker walls. The contrast creates visible annual rings. A ring is not simply a timestamp. Its width and anatomy contain information about the conditions under which it formed. If water was scarce during the period when growth was most sensitive, a ring may be narrow. Favorable moisture and temperature may produce wider growth. Fire can leave scars. Insect outbreaks can alter growth across many trees. Volcanic eruptions can influence climate strongly enough to appear indirectly in tree ring patterns. Dendrochronologists compare overlapping ring sequences among living trees, historical wood, archaeological timbers, and preserved dead trunks. If a distinctive pattern of wide and narrow rings appears in multiple samples, the sequences can be aligned like barcodes. This cross-dating allows individual rings to be assigned to exact calendar years. Tree ring records have become one of science's most precise archives of past environments. They help reconstruct drought, temperature, fire history, glacier change, river flow, and other variables. They have also been used to calibrate radiocarbon dating because the age of a particular ring can be known independently. A tree, without intending to record anything, can preserve a year-by-year history in its structure. Not every tropical tree forms one obvious annual ring, because rainfall and growth seasons can be irregular. Not every ring responds to climate in the same way. A ring pattern must be interpreted using the ecology of the species and the site. But in the right places, a trunk becomes an archive written by cell division. Wood also solves an engineering problem. As trees rise, they must support increasing weight and resist wind. Lignin strengthens cell walls and makes them more resistant to compression. Cellulose microfibrils contribute tensile strength. The arrangement of fibers, vessels, and tracheids creates a composite material that is light enough to build in enormous quantities and strong enough to hold a crown high in moving air. Trees do not simply add support everywhere equally. Mechanical stress influences growth. A leaning conifer can form compression wood. Many broadleaf trees form tension wood on the opposite side. Branch junctions thicken. Trunks exposed to wind change their allocation. The body records force as altered development. And because the growing tissues remain near branch tips, root tips, buds, and cambium, much of an old tree can be made of cells that are no longer alive. The conducting elements of mature xylem are dead at functional maturity. Their empty walls become pipes for water. Heartwood in the center of many trees no longer conducts water at all. It can accumulate resins, tannins, gums, and other compounds that slow decay. A living tree is therefore a strange mixture of active tissue and carefully preserved dead structure. This partly explains how plants can become so old. In animals, the body often depends on maintaining the integrated function of long-lived organs that cannot simply be replaced. Plants repeatedly create new modules. A branch can die while the rest survives. Roots can be lost and replaced. Bark can close over wounds. New vascular tissue can grow around damaged regions. Dormant buds can activate years later. This does not make trees immortal. Pathogens, drought, fire, wind, insects, competition, lightning, and internal hydraulic failure can kill them. But modular construction provides unusual routes around damage. Great Basin bristlecone pines are among the most famous examples. They grow high in dry, cold mountains of the western United States, often in landscapes where conditions seem too harsh for great age. Growth is extremely slow. Wood is dense and resinous. Sparse vegetation reduces competition and can limit the spread of fire. Some living bristlecone pines have been dated to nearly 5,000 years, and one tree measured in the White Mountains has exceeded 5,000 years in age. Their exact identities and locations are often protected. The familiar tree called Methuselah is therefore better described as one of the oldest famous living non-clonal trees, not simply the oldest plant on Earth. Clonal plants complicate the idea of age even more. A quaking aspen grove can consist of thousands of trunks connected to one root system and sharing the same genotype. Each visible trunk may live only a century or two, while the clone persists by continually producing new stems. Pando, the enormous aspen clone in Utah, covers more than 40 hectares and contains tens of thousands of stems. Genetic evidence confirms that much of the grove is one individual clone. Its age, however, cannot be counted the way we count rings in one trunk. The root system does not preserve an uninterrupted readable record from its first seedling. Estimates of Pando's age have ranged from thousands to much larger numbers depending on assumptions about spread, climate, and past disturbance. Claims that it is exactly 80,000 years old, or exactly 1,000,000 years old, should therefore be treated as estimates rather than measurements. The organism forces a philosophical question with a biological answer that remains practical rather than absolute. What counts as the individual? Is it one stem? One root system? One genotype persisting through thousands of replacement stems? Seagrasses create similar puzzles. Some species spread through underground rhizomes across huge areas. Genetic studies have identified extraordinarily large clones, and modeling has suggested that certain Mediterranean clones could have persisted for tens of thousands of years or potentially longer. Again, genetic size can be measured more directly than exact age. The oldest sounding numbers often depend on estimated rates of clonal spread. Plants make identity difficult because they can survive by replacing parts of themselves. They also make size difficult. The tallest living trees are coast redwoods. The largest single-stem trees by volume are giant sequoias such as General Sherman. The most massive clonal organism may be a fungus rather than a plant, depending on how mass and individuality are defined. The largest plant clone can be discussed by area, mass, stem number, or genetic continuity. Nature rarely organizes itself for record books. Each record measures one property. The deeper wonder is the range of solutions. A bristlecone survives by growing slowly in a harsh place. A redwood survives by becoming tall in a moist coastal climate. A sequoia becomes massive and fire-adapted. An aspen survives through a root system that continually replaces stems. A seagrass spreads horizontally beneath the sea. Life does not have one route to longevity. The same diversity appears at much smaller scales in leaves. Leaves may last a few weeks or many years. A deciduous tree builds a seasonal canopy and then abandons it. An evergreen keeps leaves longer, but even an evergreen replaces them eventually. Why throw away a leaf after investing resources in making it? In cold or dry seasons, maintaining broad, thin tissues can cost more than they return. Freezing damages cells and disrupts water transport. Winter sunlight may be available when liquid water is not. Drought can make every open stoma dangerous. Before a deciduous leaf falls, the tree recovers valuable nutrients from it. Chlorophyll is dismantled. Nitrogen and other compounds are transported out. As green pigment disappears, yellow and orange carotenoids that were already present become visible. Red and purple autumn colors often involve anthocyanins synthesized during senescence. Their exact adaptive roles vary among species and remain an area of research, but photoprotection is one important function proposed for some leaves while nutrients are being recovered. At the base of the leaf stalk, an abscission zone develops. Cells separate in a controlled layer. Eventually, wind or gravity removes the leaf, and a protective scar seals the stem. A forest in autumn, therefore, looks like a mass death of foliage, but it is also a carefully regulated recycling event. The fallen leaves continue the process on the ground. Fungi and bacteria decompose them. Invertebrates shred them. Carbon returns partly to the atmosphere through respiration and partly to soil organic matter. Nitrogen, phosphorus, and minerals become available again. A forest is not built only by living trees. It is built by the continual movement of matter through living and dead forms. Some of Earth's most important carbon stores exist where decomposition is unusually slow. Peatlands cover only a small fraction of the planet's land area, yet store an enormous quantity of carbon. In many northern peatlands, sphagnum mosses help create waterlogged, acidic, oxygen-poor conditions. Dead plant material accumulates faster than microbes can fully decompose it. Layer by layer, peat builds. Some peat deposits preserve thousands of years of pollen, plant fragments, charcoal, and atmospheric material. They are ecosystems and archives at the same time. Sphagnum itself has remarkable properties. Its tissues contain large dead cells that hold water like tiny reservoirs. The moss can influence acidity and hydrology, helping maintain the wet environment in which it thrives. Bryophytes are often described as simple plants because they lack the vascular systems of trees and flowering plants. That word can mislead. Their life cycles are complex. Their tissues are highly adapted to small scale. Many tolerate drying and rehydrate when water returns. Moss communities influence soil formation, temperature, nutrient cycling, and water retention across enormous areas. A moss only a few centimeters tall can help control the carbon balance of a landscape. The carbon story leads back to photosynthesis, but now at planetary scale. Every year, terrestrial plants fix vast amounts of carbon dioxide. Much of that carbon returns relatively quickly through plant respiration, microbial decomposition, fire, and consumption by animals. Some remain stored for years, centuries, or longer in wood and soil. Forests therefore participate in climate not simply by existing as piles of carbon, but through continuous flux. A growing tree takes in carbon dioxide and adds carbon to tissues. A mature forest continues photosynthesis while also losing carbon through respiration and decomposition. Disturbance can release stored carbon rapidly. Regrowth can recapture some over time. Rising atmospheric carbon dioxide can increase photosynthesis in many plants, a phenomenon called carbon dioxide fertilization, but the effect is limited by nutrients, water, temperature, species, and ecosystem structure. More carbon dioxide does not remove the consequences of heat, drought, fire, nutrient shortage, or changing rainfall. Plants respond to the whole environment, not one gas in isolation. Their response to climate is already visible in flowering dates, growing seasons, species ranges, drought mortality, fire regimes, and the timing of ecological relationships. A flower can bloom earlier in a warm spring, but its pollinator may not shift by exactly the same amount. A tree may grow faster under warmer conditions until water becomes limiting. A species may move uphill only to run out of mountain. Because plants cannot relocate as individuals when conditions change quickly, migration occurs through seeds and generations. That can be slow. Yet plants have survived enormous climatic shifts before through migration, adaptation, extinction, and replacement. Fossil pollen shows forests moving across continents after ice ages. Species that once grew far south expanded northward as glaciers retreated. The future will also involve movement, but the present rate of warming and the fragmentation of habitats create new constraints. This is one reason seed banks and botanical collections matter. A seed bank stores genetic material from plant populations under controlled conditions, often at low humidity and temperature. Not every species produces seeds that tolerate drying and freezing, so some require living collections, tissue culture, or cryopreservation. The aim is not to keep nature permanently inside vaults. It is to preserve options. A tiny seed can contain genetic combinations that no longer exist if the population disappears from the wild. And in that seed is another of the quietest forms of plant time, an organism waiting without growing. A seed contains a future plant, but it does not contain a tiny preformed tree folded inside. Plant development remains unusually open. In most animals, the major body plan is established early. Limbs, organs, and axes form during embryonic development, and later growth largely enlarges or matures structures already specified. Plants continue making new organs throughout life. At the tips of shoots and roots lie meristems, regions of dividing cells whose descendants can become many different tissues. A shoot apical meristem may continue producing leaves, stems, branches, and eventually flowers for years. Lateral meristems, such as vascular cambium, add thickness. Dormant meristems can wait inside buds until conditions change. The adult architecture is therefore partly constructed after the plant has already begun living in its environment. A tree does not know as a seed where every future branch will be. Light, damage, gravity, wind, neighboring vegetation, water, pruning, and internal hormonal relationships help determine what develops. This flexibility is visible in a familiar gardening practice. Remove the tip of many shoots and lateral buds begin growing more strongly. The apical tip had been helping suppress those buds through hormonal interactions involving auxin, strigolactones, cytokinins, and sugar availability. Once the balance changes, branches that were present only as small dormant possibilities become active. The plant rearranges its future shape. Roots do something similar below ground. Lateral roots arise from internal tissues and emerge through outer layers. Their positions and growth rates respond to water and nutrients. If one region becomes rich in nitrate, branching can increase locally. If another becomes dry, growth may be reduced or redirected. A plant can therefore invest in the part of its body that currently has the best chance of obtaining a limiting resource. Leaves also change before they even unfold. Leaves formed in bright light can differ anatomically from leaves formed in shade on the same plant. Sun leaves are often thicker, with different photosynthetic capacities and stomatal densities. Shade leaves may be broader and thinner, optimized for capturing limited light at lower construction cost. One organism can build different versions of the same organ. This capacity is called phenotypic plasticity. Plasticity does not mean that a plant can become anything. Genes define the range of possible responses. Developmental history constrains what can still change. But within those limits, one genotype can produce different forms in different environments. The appearance of a plant is therefore a conversation between inheritance and circumstance. Sometimes that conversation leaves astonishing geometries. The arrangement of leaves around a stem, called phyllotaxis, often creates spirals. New leaf primordia emerge where local hormone patterns and physical constraints allow them. In many species, the angles between successive organs produce arrangements related to Fibonacci numbers and the golden angle. Sunflower heads display intersecting spirals because florets are packed efficiently as the growing center adds new organs. There is no numerical intention inside the plant. Repeated local rules produce a global pattern. Similar self-organization shapes branching veins, root systems, flower heads, and vascular tissues. Development can be beautiful precisely because no cell sees the whole design. Each cell responds to neighbors, hormones, mechanical stress, and gene regulation. The larger form emerges. Plants also keep time. Even if a plant is placed under constant light or constant darkness, many physiological processes continue oscillating with a period close to 24 hours. Leaf movements, gene expression, hormone levels, stomatal behavior, and growth can all show circadian rhythms. The internal clock is built from networks of genes and proteins that activate and repress one another in cycles. Light and temperature reset, or entrain, the clock so that internal time remains aligned with the external day. This allows anticipation. A plant does not need to wait for sunrise before preparing photosynthetic machinery. Gene expression can begin changing before dawn because the clock predicts that light is likely to return. Stomata in many C3 and C4 plants begin opening around morning under the influence of both light and circadian signals. Enzymes and transport systems are adjusted. Growth may peak at particular times of night or day. The clock also helps measure seasons. Photoperiodic flowering depends not simply on the amount of light, but on when light occurs relative to internal circadian phases. A flash of light in the middle of a long night can alter flowering in some species because the plant's clock interprets the interruption differently from the same amount of light during daytime. Plants live inside repeated cycles, day and night, tides in some coastal species, wet and dry seasons, winter and summer. Their biology predicts recurring change. The evening behavior of flowers provides especially gentle examples. Some flowers close at night, a movement known broadly as nyctinasty. Others do the opposite and open after sunset. Tulips, crocuses, and many daisy-like flowers alter petal position as light or temperature changes. Legume leaves may fold into nighttime positions through water pressure changes in pulvini. Some plants raise leaves, others lower them. These movements can reduce heat loss, protect reproductive structures from dew or rain, change herbivore exposure, or match the activity of pollinators. Their adaptive significance varies. Night-blooming plants synchronize scent and nectar with nocturnal visitors. A moonflower or night-blooming jasmine can increase fragrance after sunset. Moths searching in darkness follow odor plumes. Pale petals reflect available light. Some cactus flowers open for only one night, investing enormous resources in a brief window when bats or moths are active. The flower schedule is part of its anatomy. At night, photosynthesis stops in ordinary green tissues because there is no light to drive the photosynthetic electron reactions. Respiration continues. Plants respire all the time, day and night. Mitochondria break down organic molecules and use oxygen to produce ATP. During the day, photosynthesis usually dominates gas exchange in illuminated leaves, so the net effect is carbon dioxide uptake and oxygen release. At night, without photosynthetic oxygen production, respiration becomes more visible in the balance. Oxygen is consumed and carbon dioxide is released. This is sometimes used to create unnecessary anxiety about sleeping near houseplants. The amount of oxygen consumed by ordinary houseplants at night is tiny compared with the air in a room and the respiration of people and animals. A plant on a bedside table is not meaningfully stealing your oxygen. The important point is simply that plants are living organisms, not solar machines that turn off in darkness. Their cells still require energy. Stored starch made during the day can be broken down at night. Many plants regulate this use carefully enough that reserves last until dawn. In Arabidopsis, a model plant used extensively in research, starch consumption during the night adjusts to the expected time remaining before morning. The details are molecular, but the effect is almost elegant enough to sound planned. The plant avoids spending tomorrow's dawn energy at midnight. Growth can continue in darkness. In some shoots, cell expansion is particularly active at night because water status is favorable and sugars remain available. Roots continue growing. Phloem continues transporting carbon. Hormonal signals continue moving. Proteins are repaired and replaced. DNA is transcribed. Cells divide. And, as we have already seen, CM plants may open stomata at night and actively take in carbon dioxide. A desert at midnight can therefore contain plants performing one of the key steps of photosynthesis, while their stomata would be more tightly closed under the heat of noon. The same night can mean different things to different leaves. Seasonal darkness matters too. In temperate forests, winter buds are extraordinary packages of suspended growth. Many are formed months before they open. Tiny embryonic leaves or flowers are enclosed by protective bud scales. Hormonal changes induce dormancy. Cold hardiness increases. Water content and cellular chemistry change to reduce freezing damage. Some plants produce antifreeze like proteins or accumulate sugars and other solutes that help stabilize membranes. Ice may form in extracellular spaces, while sensitive cell interiors remain protected from lethal ice crystals. A bare winter tree looks empty. Its next spring may already be physically present in miniature at the ends of its branches. Dormancy protects future growth from a warm week arriving too early. Many buds require a certain amount of chilling before they can respond fully to later warmth. This chilling requirement makes seasonal timing more reliable. Climate warming can complicate the system. Winters may become warm enough to reduce chilling, while springs also arrive earlier, shifting bud burst in complex ways. The plant measures winter not with one thermometer reading, but through accumulated physiological history. Time also appears in roots. Some root systems undergo seasonal pulses. Fine roots are produced and die. Fungal partners change in abundance. Stored carbohydrates move between roots and shoots. Trees that appear inactive above ground may still maintain substantial metabolism beneath frozen or cool soil when conditions permit. The world under snow can remain biologically active. Snow itself can insulate soil, keeping it warmer than exposed winter air. Microbes continue slow decomposition. Roots may remain unfrozen below the surface. Mycorrhizal hyphae persist. A forest does not stop when leaves fall. It changes the location and speed of its work. The same principle is true across the entire plant kingdom. Desert annuals spend much of the year as seeds. Deciduous trees withdraw resources into wood and roots. Evergreen conifers keep leaves, but reduce photosynthetic activity under severe cold. Tropical forests may respond more to rainfall than temperature. Mediterranean plants may slow during summer drought and grow during cooler wet seasons. There is no universal plant calendar. Each lineage has evolved around recurring patterns in its own environment. Those patterns can become so precise that a plant's presence reveals the ecology of a place. Mangroves tolerate salt and waterlogged, oxygen-poor sediments. Some exclude much of the salt at their roots. Others secrete salt through leaves or store it in tissues that are later shed. Their specialized roots can provide stability in soft mud and improve gas exchange. Alpine cushion plants grow low to the ground, creating compact forms that reduce wind exposure and trap warmer air. Their shape modifies the microclimate for themselves and sometimes for other organisms nearby. Resurrection plants can survive extreme drying. Their tissues lose most free water, metabolism nearly stops, and protective sugars and proteins help stabilize membranes and cellular structures. When rain returns, they rehydrate and resume activity. Salt-tolerant halophytes manage ion concentrations that would damage ordinary plants. Some compartmentalize salt inside vacuoles. Some develop succulent tissues that dilute salts with water. Aquatic plants have moved back into water after their ancestors spent millions of years adapting to land. Seagrasses evolved from flowering plants that return to the sea. They pollinate underwater, form meadows, stabilize sediments, and provide habitat for fish, turtles, invertebrates, and microorganisms. Evolution is not a ladder moving from water to land toward ever greater complexity. It is branching experimentation. A lineage can return to an environment its distant ancestors left and build a new set of adaptations from the structures it already possesses. Humans entered this branching history as one more animal dependent on plants, then began reshaping it unusually rapidly. For most of our species' existence, people gathered wild plants and managed landscapes in diverse ways. Thousands of years ago in several regions, communities began domesticating particular species more intensively. Seeds were saved from plants with useful traits. Generation after generation, human preference became a powerful selective force. Wild grasses became wheat, rice, maize, barley, millet, and other staples. Teosinte, the wild ancestor of maize, bears small, hard fruiting structures that look very different from a modern ear of corn. Artificial selection changed branching, seed number, seed exposure, and many other traits. While Brassica oleracea gave rise to cabbages, kale, broccoli, cauliflower, Brussels sprouts, and kohlrabi through selection on different parts of the same species. A single plant genome can contain developmental possibilities that humans amplify in very different directions. Domestication often reduced natural seed dispersal. A wild grain benefits if mature seeds fall away from the parent. A farmer benefits if seeds remain attached until harvest. Mutations causing non-shattering seed heads therefore became favored under cultivation. Traits that would be disadvantageous without people could become successful because people carried, protected, watered, and replanted them. Agriculture transformed both plants and humans. Permanent food production supported larger settlements in many regions, though the history was neither simple nor uniform. Diets changed. Landscapes were cleared, irrigated, burned, terraced, and fertilized. Crop diseases influenced history. Trade routes moved seeds across continents. Today, a large fraction of human calories comes from a surprisingly small number of plant species. Yet crop wild relatives and traditional varieties contain genetic diversity that may become crucial as climates, diseases, and pests change. A gene for drought tolerance may be unimportant in one century and essential in another. Plant diversity is therefore not merely a catalog of beautiful forms. It is a library of evolutionary experiments, many of which we have barely examined. And that library is still incomplete. Scientists continue describing thousands of plant species. Many more are thought to remain unnamed, especially in biodiversity-rich regions. Some may disappear before they are formally documented. A species can be globally rare and locally ordinary to the few people who live beside it. Botanical knowledge exists not only in herbaria and laboratories, but in indigenous and local traditions developed through long relationships with particular landscapes. Modern plant science increasingly combines genetics, remote sensing, physiology, ecology, fossils, collections, and field observation. Satellites measure greenness across continents. Fluorescence from chlorophyll can provide information about photosynthetic activity. DNA reveals relationships invisible from appearance. Isotope ratios trace water and carbon. Tiny sensors measure sap flow. Microscopes watch stomata move. Sequencing reveals which genes activate during drought or infection. Yet some of the most important plant observations remain simple. When did the first leaf unfold? When did flowering begin? Which pollinator arrived? How deep did the roots grow? Which seed survived? How wide was this year's ring? Science can begin with noticing what was always happening slowly enough to be missed. One of the reasons plant biology is so easy to exaggerate is that new instruments reveal signals that were once invisible. Electrical activity is a good example. Animal nerves use rapid electrical changes across cell membranes to transmit information. Plants also generate electrical signals, though they do not have neurons or a nervous system. When a Venus flytrap trigger hair bends, ion channels open and an action potential can spread across the trap. In wounded leaves, electrical and calcium signals can travel away from the damaged site and contribute to defensive responses elsewhere. Hydraulic pressure changes and chemical signals interact with these electrical events. Calling this a plant nervous system would blur important differences. The fascinating point is that excitable membranes evolved uses far beyond animal brains. Living cells everywhere maintain voltage differences because ions are distributed unequally across membranes. Evolution can turn those gradients into signals in many ways. Plants also experience vibration. Roots and stems are constantly exposed to mechanical waves from wind, rain, insects, flowing water, and movement in soil. Experiments have found that plants can alter growth or defense after particular vibrations, and some studies suggest responses to frequencies associated with pollinators or herbivores. This is sometimes summarized by saying that plants can hear. The shorthand is memorable, but it can hide the mechanism. A plant has no ear, auditory nerve, or sound perception resembling ours. Mechanical vibration can deform membranes and cell walls, activate mechanosensitive channels, and change signaling. The relevant biology is mechanosensing, living tissue extracting information from physical movement. The same precision helps with words such as memory, counting, and decision. Plants can retain information from earlier events without recalling an experience. Vernalization leaves molecular changes after winter cold. Immune priming changes later responses. Drought can alter gene expression after soil rehydrates. A Venus flytrap integrates repeated touches across a short interval. Circadian clocks use previous cycles to anticipate dawn. That history can be stored in proteins, hormones, chromatin states, metabolites, and altered anatomy. The response is real even when the psychological metaphor is removed. Take a young sunflower turning across a day. The motion is produced by differential growth and changing orientation, coordinated by light sensing and circadian regulation. Young sunflower plants and developing flower heads can track the sun from east to west during the day and reorient eastward at night. As flower heads mature, the tracking largely stops and they tend to face east. That eastward orientation can warm flowers earlier in the morning and influence pollinator visits. No flower has watched the sky. Millions of cells have changed growth according to local molecular signals. and the combined result is a movement synchronized with a star 150 million kilometers away. Or consider a pine cone. The scales of many cones open in dry conditions and close when moist, even after the cone tissue is dead. Different layers of the scale swell by different amounts as humidity changes. The structure itself converts environmental moisture into movement without metabolism. A dead plant structure can therefore remain mechanically responsive. Some seed pods twist as they dry because layers of cellulose fibers are arranged in different directions. The drying tissue stores elastic energy until a threshold is crossed and the pod splits. Some grasses drill their seeds into soil with hygroscopic awns that twist and untwist as humidity changes. In these examples, there is no signaling network at all. Material properties perform the work. Plants are full of such passive mechanisms because evolution can exploit physics instead of spending metabolic energy. Color is another place where chemistry and physics meet. Chlorophyll makes many tissues green, but plants produce pigments across the visible spectrum. Carotenoids create yellows and oranges and also protect photosynthetic machinery by dissipating excess energy. Anthocyanins produce reds, purples, and blues whose exact color depends on cellular conditions. Betalains color certain plant families. Structural features on petal surfaces can scatter light, alter gloss, or create iridescence. A flower's color is not simply paint applied to tissue. Pigments are dissolved or bound in cellular compartments. Cell shape changes how light enters and exits. Tiny conical epidermal cells can intensify color and provide grip for pollinating insects. Some petals contain microscopic structures that produce optical effects independent of pigment, much as feathers or butterfly wings can. A bee approaching such a flower receives a visual signal shaped by molecules and geometry together. Leaves use geometry too. Many forest leaves have drip tips that help water run from the surface. Desert leaves may be small, rolled, hairy, waxy, or reflective, reducing heat load and water loss. Succulents store water in thick tissues. Needles reduce surface area and tolerate cold and drought. The enormous leaves of tropical plants and the tiny needles of alpine conifers are not degrees on a scale from primitive to advanced. They are solutions to different balances of light, heat, water, wind, and construction cost. Even leaf lifespan reflects economics. A thin, fast-growing leaf may capture carbon quickly, but be vulnerable and short-lived. A thick evergreen leaf can be expensive to build yet survive for years in nutrient-poor or stressful environments. Ecologists describe suites of such traits as part of a leaf economic spectrum. Plants invest resources as if they faced budgets because they do. Every gram of carbon and nitrogen placed into one structure cannot simultaneously be placed into another. The Frey strategy is useful as long as we remember that natural selection, not foresight, produced the pattern. The same resource trade-off shape forests. A fast-growing pioneer tree may colonize bright, disturbed ground, producing low-density wood and abundant leaves. A shade-tolerant species may survive for years beneath a canopy, growing slowly until a gap appears. Seeds can wait in soil. Seedlings can wait in shade. Adult trees can wait through seasons. Then a storm opens the canopy. Within hours, light levels on the forest floor change. Soil temperature shifts. Rain reaches the ground differently. Dormant buds respond. Existing seedlings accelerate photosynthesis. New seeds germinate. A forest is continually being rebuilt through gaps. Large disturbances operate at larger scales. Fire is destructive, but in fire-adapted ecosystems, it is also an ancient ecological process. Some pines hold seeds in cones sealed by resin until heat opens them. Some shrubs resprout from underground lignotubers or protected buds after their stems burn. Smoke chemicals stimulate germination in certain species. thick bark protects cambium in fire-tolerant trees. Other plants are poorly adapted to fire and die readily. The ecological effect depends on fire frequency, intensity, season, fuel, and the evolutionary history of the community. Suppress fire for long enough in an ecosystem adapted to frequent mild burning, and fuel structure may change. Increase fire frequency beyond the recovery capacity of a forest, and trees may fail to return. There is no single meaning of fire for plants. The same event can be renewal in one landscape and catastrophic conversion in another. Flooding offers another contrast. Roots need oxygen for respiration. In waterlogged soil, gas diffusion is slow and oxygen can disappear rapidly as microbes and roots consume it. Many ordinary plants suffocate. Wetland plants evolve solutions. Parenchyma creates internal air spaces that move gases between shoots and submerged tissues. Mangroves produce specialized above-ground or shallow roots in some species. Flood-tolerant plants alter metabolism and hormone signaling. Rice can survive conditions that would kill many crops because its anatomy and physiology are adapted to water. Some rice varieties elongate stems rapidly during deep flooding, trying to keep leaves above the surface. Others enter a low-growth survival state under complete submergence, conserving carbohydrates until floodwater recedes. Two opposite responses can solve different versions of the same problem. When drought arrives instead, the challenge reverses. Roots may explore deeper soil. Stomata close. Leaf expansion slows. Abscisic acid signaling increases. Osmotically active compounds help cells retain water. Some species shed leaves. Others tolerate extremely low tissue water potentials. Drought resistance can mean avoiding water loss, finding more water, storing water, tolerating dehydration, or escaping the drought entirely by completing a life cycle before it begins. A desert annual may spend 11 months as a seed and a few weeks as a flowering plant. From the seed's perspective, the harsh season is not something to endure as an adult. It is something to sleep through. That word, sleep, brings us close to the quiet mood of tonight. Plants do not sleep as animals do. They do not pass through REM and non-REM stages. They do not need unconsciousness, but they do have nights. They reduce some processes, increase others, redistribute resources, adjust posture, open or close flowers, alter hormone levels, and follow internal clocks. Darkness is a biological condition, not an absence of biology. And because the night changes plants, plants also change the night around them. Step outside into a garden after sunset, and most of what reaches you will still be evidence of plant activity. The air may carry floral volatiles released specifically at night. Moisture may collect on leaves. The temperature near vegetation may differ from that over pavement because plants change the daytime balance of radiation and water. In a forest, trunks and leaves slow wind. Roots alter drainage. Fallen litter insulates soil. Canopies intercept rain. Transpiration during the day has moved water back into the atmosphere, where it may later contribute to clouds and rainfall far away. Plants create habitat partly by being bodies. A tree is vertical geography. Its bark holds lichens, mosses, fungi, bacteria, insects, spiders, and eggs. Cavities shelter birds and mammals. Leaves host caterpillars. Flowers feed pollinators. Fruits feed dispersers. Roots surround microbial communities. Deadwood becomes habitat for organisms that cannot live in healthy wood. When the tree eventually falls, the habitat does not end. A fallen trunk stores moisture and carbon. Fungi digest lignin and cellulose. Beetle larvae tunnel through it. Mosses spread across the surface. Seedlings establish in the decaying wood. Nutrients move back into soil. Death converts architecture into substrate. This cycling is one reason the opening statement that plants support life is more than a metaphor, but it also deserves precision. On land, Plants are the dominant primary producers in most ecosystems. They capture light energy and convert inorganic carbon into organic matter that feeds herbivores, decomposers, parasites, and predators through long chains of exchange. But plants are not Earth's only photosynthetic organisms. Algae and cyanobacteria, especially in the oceans, make enormous contributions to global primary production and oxygen cycling. The atmosphere you are breathing is the legacy of photosynthesis across the biosphere over geological time, not the work of one tree outside the window. Still, terrestrial life is profoundly organized around plants. A deer eating grass is using carbon fixed by chloroplasts. A wolf eating the deer is using that same chemical energy one step later. A fungus decomposing a branch is using compounds built from atmospheric carbon dioxide. Your own meal may contain plant tissue directly or may contain animal tissue ultimately supported by plant or algal production. Even fossil fuels are ancient biological carbon transformed over immense time, though burning them returns carbon to the atmosphere far faster than present ecosystems can simply absorb without consequence. Plants sit at an intersection between atmosphere, soil, sunlight, and life. Their roots bring mineral nutrients into biological circulation. Their leaves move water to the air. Their tissues store carbon. Their litter builds soils. Their flowers and fruits restructure animal behavior. Their evolution has changed atmospheric chemistry and landscapes. And yet no individual plant performs any of this for the planet. A leaf opens because local development produced it. A stoma responds because guard cells change their ion balance. A root branch forms because signals favor growth there. A flower attracts an insect because ancestors with effective attraction left more offspring. Planetary effects emerge from countless local processes. This is perhaps the quietest wonder of plants. There is no forest-wide command center deciding to make rain, store carbon, feed insects, and build soil. There are cells following molecular rules. Scale them across billions of leaves and thousands of years, and they become climate, habitat, food webs, and landscapes. The same idea appears inside one plant. No cell understands the tree. A root cell senses gravity and water. A guard cell responds to light and abscisic acid. A cambial cell divides. A flower meristem activates a developmental program. A companion cell loads sugar into phloem. The tree emerges from communication among parts that each possess only local information. Plants are distributed organisms in a very literal sense. This distribution creates resilience, but also unusual vulnerabilities. A grazing animal can remove half the leaves without necessarily killing the plant. A branch can break and be replaced. Yet damage to a narrow ring of living tissue around a trunk can be fatal if it interrupts phloem around the entire circumference. A pathogen that reaches vascular tissue can spread rapidly. Drought that breaks too many xylem pathways can cause whole branches to fail. Resilience is never invulnerability. Plants survive through trade-offs. A thick leaf resists stress but costs more carbon. A wide xylem vessel moves water efficiently but can carry hydraulic risks. An open stoma gains carbon and loses water. A fragrant flower attracts pollinators and may attract herbivores. A sweet fruit feeds a disperser and consumes resources that could have made more seeds. Evolution does not remove these conflicts. It finds workable compromises. That is why plant diversity is so large. There is no one perfect leaf because a perfect leaf for a cloud forest would fail in a desert. There is no one perfect root system because shallow wet soil and deep dry soil reward different architectures. There is no one perfect flowering time, seed size, wood density, or lifespan. Every species is an answer to a particular history of constraints. Some answers are temporary. Environments change. Competitors arrive. Pollinators disappear. Pathogens evolve. Continents move. Climates shift. The fossil record is filled with plant lineages that once dominated landscapes and later declined or vanished. Entire forests of giant lycophytes and horsetail relatives covered carboniferous wetlands. Their dead tissues contributed to coal deposits that humans would burn hundreds of millions of years later. Later worlds were dominated by gymnosperms. Flowering plants transformed ecosystems again. Today's forests are not the final form of vegetation. They are one frame in a much longer film. If time could be accelerated, the apparent stillness would disappear. Seeds would rise from soil like small green waves. Branches would spread into openings in the canopy. Vines would circle supports. Flowers would open and collapse. Leaves would turn, age, and fall. Trunks would thicken. Roots would move beyond the frame. Accelerate further, and whole forests would shift across continents as climates changed. Species would appear, diversify, retreat, and vanish. Then slow time back down to tonight. All that movement becomes quiet again. Perhaps this is why plants fit so naturally into a bedtime story. Their biology contains enormous activity without the kind of visible urgency that attracts an animal's attention. Molecular events are fast. Electrons move. Enzymes react. Membranes change voltage. But the consequences we can see often unfold as growth, accumulation, season, recovery, and waiting. Consider an old tree in winter. From a distance, it may seem almost inactive. A deciduous crown has no leaves. Shoot growth has stopped. The branches are still. Yet living cells beneath the bark continue metabolism. Stored carbohydrates remain in wood and roots. Buds already contain future tissues. Protective chemistry reduces freezing injury. Roots and soil organisms continue some activity whenever temperature and moisture permit. When spring arrives, the sudden swelling of buds is only the visible phase of a process prepared long before. Many of those leaves began forming inside buds during the previous growing season. Winter provided chilling. Day length changed. Warmth accumulated. Hormonal balances shifted. The tree did not begin again from nothing. It carried its future quietly through the cold. A similar change of pace is happening tonight in plants around you. Many C3 and C4 plants have narrowed or closed their stomata in darkness. Some CM plants are doing the opposite, opening theirs and taking in carbon dioxide while the air is cooler. Some flowers have folded. Others have only just opened for moths or bats. Starch made during the day is being used through the hours before dawn. Roots continue absorbing water and ions. Fungal hyphae continue through soil. Mitochondria keep producing ATP. Cells and meristems maintain the tissues from which future growth will come. The night does not switch plant life off. It changes the schedule. And below the soil, darkness is not even a change. Roots spend most of their existence there. They respond to gravity, moisture, nutrients, oxygen, pressure, microbes, and obstacles without ever seeing daylight. Above them, leaves are built around light. The two worlds are connected by narrow vascular strands. Water moves from the darkness of soil toward illuminated leaves. Carbon fixed in those leaves returns through phloem toward roots, buds, fruits, fungi, and growing tissues. A root does not need to see the sun to depend on it. A leaf does not need to touch soil to depend on what roots obtain there. The plant holds those distant environments together, and its materials rarely remain in one place. Carbon dioxide from air becomes sugar. Some carbon becomes cellulose in a stem. Some becomes nectar carried away by an insect. Some enters fruit eaten by a bird. Some travels into roots and fungal partners. Some falls with a leaf and becomes part of soil before eventually returning to the atmosphere. Water moves too. Minerals move. Hormones move. Electrical signals move. Pollen and seeds carry genes into new places. Over geological time, forests themselves move as generations establish farther north, south, uphill, downhill, inland, or toward coasts. Plant life is movement made difficult to notice by scale. So tomorrow, when you pass a tree, a weed in a crack, a patch of grass, or a plant on a windowsill, there is no need to imagine a hidden mind inside it. The real biology is stranger enough. A body without muscles changes position. A body without a heart can lift water more than 100 meters above the ground. A body without a brain can combine signals from light, gravity, water, touch, pathogens, hormones, and its own internal clock. A seed can remain almost motionless while preserving the possibility of an entire future plant. A tree can preserve drought and rainfall in the width of rings. A flower can exploit wavelengths a bee sees and humans do not. A root can change the chemistry of the soil immediately around it. A desert succulent can wait for darkness before opening its pores to the atmosphere. None of this requires plants to be miniature animals. It requires only the extraordinary solutions that evolution has built from cells, chemistry, physics, and time. And now, somewhere outside, a leaf is in darkness. Its photosystems are no longer receiving the stream of photons they used during the day. But the leaf is not empty or inactive. Sugars are being transported and respired. Membranes maintain gradients. The circadian clock continues toward dawn. Farther down, a root tip may be extending by a fraction of a millimeter around a grain of soil. Nearby, a fungal filament may be growing through a pore too small for that root to enter. A dormant seed may remain almost completely still. That stillness is also a biological achievement, to wait without losing the possibility of beginning. One flower may be closed. Another may be releasing scent because its pollinator is awake now. There is no universal plant night, only many evolutionary histories entering the same darkness in different ways. You are connected to those histories without needing to think about them. Food, wood, fibers, oils, medicines, fruits, grains, spices, coffee, tea, and countless other materials come directly from plant bodies or from chemistry plants evolved. And the oxygen cycle that supports animal respiration depends on photosynthesis across land and oceans over immense stretches of time. None of it needs supervision tonight. No root needs to be told which way gravity lies. No seed needs encouragement to wait. No tree needs attention in order to move water. And when morning comes, photons will cross the distance from the sun again. Chlorophyll will absorb some of them. Electrons will move. Water will be split. Carbon dioxide will enter leaves. Sugars will begin accumulating in the light. The process will be ancient, ordinary, and almost invisible. For now, the leaves can remain dark. The roots can remain hidden. The seeds can wait. And you can rest. Sleep well. Good night.